Inflammation involves a wide range of chemical signals. Today, we will focus on cell-derived mediators, which play a major role in both acute and chronic inflammation. What are cell-derived mediators? Cell-derived mediators are chemicals produced by inflammatory and tissue-resident cells. Some are stored inside granules and released immediately when needed, while others are newly synthesized in response to inflammatory stimuli. These mediators exert their effects by binding to specific receptors on target cells. Important cellular sources include mast cells, basophils, platelets, neutrophils, macrophages, and endothelial cells. Their wide distribution ensures that inflammation can be initiated very quickly wherever injury occurs. The major categories of cell-derived mediators include vasoactive amines, arachidonic acid metabolites, lysosomal enzymes, platelet-activating factor, cytokines and chemokines, and free radicals such as reactive oxygen species and nitric oxide. Each category contributes to different aspects of the inflammatory response. The two main vasoactive amines are histamine and serotonin. Histamine is primarily released from mast cells and causes vasodilation and increased vascular permeability, leading to symptoms like redness, warmth, and swelling. Serotonin is mainly derived from platelets and has similar but generally weaker effects. These mediators act within the first hour of inflammation, making them crucial early responders. Arachidonic acid is released from cell membranes and converted into multiple mediators through two main pathways: the CoX pathway, producing prostaglandins and thromboxane, and the LOX pathway, producing leukotrienes and lipoxins. Prostaglandins cause pain, fever, and vasodilation. Thromboxane promotes vasoconstriction and platelet aggregation. Leukotrienes cause bronchoconstriction and attract leukocytes. Lipoxins perform the opposite role. They help resolve inflammation. Neutrophils and macrophages release lysosomal enzymes to destroy pathogens. However, these enzymes can also damage host tissues if released excessively, contributing to chronic inflammation and tissue injury. PAF is produced by several cell types, including basophils, macrophages, endothelial cells, and platelets. It dramatically increases vascular permeability, even more than histamine, and also promotes platelet aggregation, bronchoconstriction, and leukocyte recruitment. Therefore, it plays a strong role in amplifying inflammation.
Cytokines such as TNF-alpha and IL-1 promote fever, endothelial activation, and leukocyte recruitment. Chemokines like IL-8 guide neutrophils to the site of injury. Interferon gamma activates macrophages to enhance microbial killing. Overall, cytokines and chemokines coordinate the movement and activity of immune cells. Reactive oxygen species produced by neutrophils and macrophages help kill microbes, but can also injure surrounding tissue and endothelium. Nitric oxide from endothelial cells and macrophages causes vasodilation and helps inhibit platelet aggregation. Both are essential components of host defense, but must be tightly controlled. To summarize, vasoactive amines initiate immediate vascular changes. Eicosanoids regulate pain, fever, and vascular tone. Lysosomal enzymes contribute to host defense but may damage tissue. PAF intensifies inflammation. Cytokines direct leukocyte recruitment and systemic reactions. And reactive oxygen species and nitric oxide help eliminate microbes. Altogether, these cell-derived mediators are central to both the beneficial and harmful effects of inflammation. Inflammation is a protective process, but when mediator release becomes excessive or prolonged, it can lead to tissue damage. Understanding these mediators helps us recognize disease mechanisms and therapeutic targets. Thank you, and feel free to ask if you have any questions.